Let's do an MCAT problem. A researcher is analyzing a family with an X-linked recessive disorder. They use a restriction enzyme that recognizes the sequence 5' GATC 3'. On the normal allele, there are two of these sites flanking a 200 base pair segment of DNA. On the disease-causing allele, a point mutation has occurred, changing one recognition site to 5' GTTC 3'. If the researcher analyzes DNA from an unaffected carrier female using a southern blot with a probe specific to this 200 base pair region, what is the expected result? A single band corresponding to a DNA fragment larger than 200 base pairs. B, two bands, one at 200 base pair and one significantly larger band. C, a single band at 200 base pairs. Or D, no band will be visible because the probe cannot bind to the mutated allele. Now, this question requires background knowledge of two separate fields of genetics. One is the Mendelian understanding that there are two X chromosomes in a unaffected carrier female. One X chromosome with the disease-causing allele and the other X chromosome that does not have the disease-causing allele. The other is the molecular biology technique of the southern blot, picking up on DNA fragments. The restriction enzyme cuts the DNA at a specific site. In this case, on the normal X chromosome, it will make two cuts, the 200 base pair fragment, and one will be the 200 base pair fragment, and the other one will be missing the cutting site, and therefore will have a longer fragment. The mutated X will not match the restriction enzyme cutting site, and therefore will leave behind a longer fragment. This produces two bands. Thank you for playing, and for personal MCAT help, I'll see you at mcatboss.com.